Welcome, everyone. Thanks for coming to the 9 a.m. session. My name is David Moore. I run the high throughput sequencing lab at Johns Hopkins Genomics/Genetic Resources Core Facility. I'd like to introduce our first speaker of the morning. Her name's Ava Hoffman. Ava is a senior staff scientist at Fred Hutch Cancer Center and a faculty associate at JHU Biostats. She serves as PI of the Data Science for Environmental Health program and is co-PI of the Anvil project. She also organizes the Genomic Data Science Community Network, exploring ways to make genomics research more accessible by connecting communities to cloud-based resources. Today, Ava will be presenting her work in this area. Our talk's entitled Digging into Genomics with BIODIGS at Under-Resourced Institutions. Please welcome Ava.
For having me. We're at the Plenary here. Okay. See if I can work this. Okay. Yes. Awesome. All right. Well, again, thanks for having me. Hopefully everyone has their coffee. I know it's pretty early, but appreciate you making it out this morning. As David said, I'm Ava Hoffman. I'm actually originally trained as an ecologist, so this project is super exciting and something I'm just really stoked to tell you all about today. So let's get into this project. It's really, really new, so you're hearing something that's hot off the press, no papers or anything like that yet. But yeah, let's talk about digging into genomics with BIODIGS at under-resourced institutions. Okay, so I probably don't need to tell you all about the importance of biodiversity in the natural world. So it's important for the functioning of ecosystems and, basically anything out in nature in terms of giving us clean water, preventing erosion, pollination, clean air, things like that. I think a lot of the times when we think about the diversity that's out there, we think about big trees and mammals and birds and things like that we can see. But I want us for a moment to imagine the world underneath our feet in the soil. It's the ecosystem that we might encounter when we pick up a little bit of dirt. So to give us an idea of what's going on in there, I want us to imagine a gram of soil, maybe something about that big. And in that gram of soil, we can have up to 10 billion bacterial cells, representing anywhere from 4,000 to 50,000 species. So it's just incredibly diverse. You can just see a tiny fraction of what you might encounter: some mites, some arthropods, we've got some cool-looking worms and nematodes, as well as phages, bacteria, fungi, all kinds of stuff. And I think this quote sums it up really nicely from Anthony et al. 2023. Said soil is likely home to 59% of life. So 59% of species, making it the singular most biodiverse habitat on Earth. It's fundamental for our fibers that we generate, for fuel, as well as for the health of ecosystems and human systems and things like that. But we are in a biodiversity crisis with climate change and the effects of humans on Earth. We're losing species a lot faster than we think we should. So it's really critical that we understand what's going on in the soil. We understand that diversity, and we're able to kind of suss it out and understand why, what it's doing and why it's important. But, thinking about the different kinds of soils we might encounter, this is going to take a huge team and a huge effort. It's certainly not something I can do alone, or even a small lab group can do alone. And so this is where the Genomic Data Science Community Network comes into the story. This is a large team of folks who are just collectively interested in promoting education and research in genomic data science, so including things like bioinformatics and methods and things like that, as well as genomics and kind of the broader ecological and health implications. At historically Black colleges, Hispanic serving institutions, tribal colleges, community colleges, and other smaller, underserved or focused institutions that may be a little less research heavy. And so this is a group that's distributed all across the country. We've got some folks in Puerto Rico and down here, and hoping to expand. This is an incredibly diverse group in terms of interests and backgrounds. We've got some physicians, folks that are interested in medicine and public health, but also folks that are interested in plant genomes and computational and biostatistic methods and things like that. We started, see our Zoom picture, we started during the pandemic and just kind of a need to connect with other like-minded folks. But we kind of kept it going, and it's been a really amazing effort and really amazing group of folks. You can see us, we actually had an in-person symposium late last year. So we've kept up the gatherings and meetings to talk about the things we're excited about. So one of the first things we did as the GDSCN was try to figure out some of the shared and common needs and resources of folks, students and faculty working at these under-resourced institutions. We put together this paper in late 2022, diversifying the genomic data science research community, where we talked about some of the needs of these faculty and their unique institutions. And I think at the highest level, we wanted to make sure that we're serving the students here. So helping them recognize the growing importance of genomic data science, genomics, and all the skills associated with it. A lot of the, the tools and skills that you learn doing genomics, like coding and things like that, are applicable even outside of the field. So helping them recognize how it could be useful for their future careers, but also helping bring light to, some of the needs of the under-resourced institution faculty. So helping support their professional development, learn the newest cutting-edge techniques, and helping them feel empowered to teach some things in genomics as well. But before we move on, I did want to call attention to this right here. So the R1 faculty are the folks at the Johns Hopkins and the Fred Hutches of the world, folks that get a lot of research funding. They're really in the weeds of applying for grants and getting funds and doing big research projects. And I think R1 faculty are actually uniquely situated to help this whole environment and support students at under-resourced institutions, especially by creating curricula on cutting edge techniques and cutting edge topics in genomics, and helping work with faculty to develop authentic research projects. That was just time and time again something that came up, was the need to get students and faculty involved in real research. Because a lot of these institutions, they don't have a big server, they don't have large lab space, they don't have a lot of those resources that we do already have set in place at the Johns Hopkins of the world. So this collaboration and resource sharing is really essential, I think, to this environment, and something that I want us to keep in mind as this talk goes forward. Okay, so highlighting that. All right, and I did want to mention too that the Genomic Data Science Community Network is expanding and growing in the past year or so. So the NIH put out a call for a computational genomics and data science hub for education and research for students at under-resourced institutions. And so our collaborator, Kristen Reinhardt, developed the Grads 4C program to kind of establish some more infrastructure along those lines. So things like workshops, symposia, really focusing on professional development for these students, and really getting them into the weeds of the data science part of things. So we're working really closely with her, and excited to see how we can kind of continue to work together to make BIODIGS really successful. Okay, so speaking of BIODIGS, I mentioned that the authentic research experiences were really important to the faculty and students. And so this is where BIODIGS was born. BIODIGS is a project, biodiversity and informatics for genomics scholars. So not only are we interested in exploring the taxonomic and genetic diversity out in the world, but also interested in the informatics side of things. So the tools, the techniques, and the methods that are going to be used in genomic science. All right, so we have a project name, but what questions are we actually going to ask? What can we actually learn from the soil? So as I mentioned, the Genomic Data Science Community Network, GDSCN, is an incredibly diverse network of faculty. They have tons of different interests. And so the soil is really a perfect place to answer a lot of different questions, a lot of different things that they're interested in. So we can ask questions about microbiology and metagenomics. So what's actually living in the soil? It's kind of hard to look unless you've got a really powerful microscope. So what's actually there? Can we assess that with a genomic approach? And how does the genomic composition change in time and space? These environments are incredibly dynamic. What happens when it rains? What happens between winter and summer? Those kind of questions. We can answer genomics and bioinformatics questions. So what are the optimal approaches for understanding the soil metagenome, metagenome assembly and classification? And are there different methods we might want to take to extract DNA and to try to get those findings ready to go and get the data ready? And are there merits and trade-offs that we might want to consider when we're thinking about short versus long read sequencing? So some of those more technical questions. There's questions about agriculture and the environment. So how do the characteristics of the soil and the soil microbiome actually affect the bigger picture? So affecting plants and animals and agricultural systems. And then finally, for a public health perspective, so how does the soil microbiome influence the human microbiome, skin, gut, all that stuff, and human health outcomes? So we know that the soil can have traces of things like heavy metals, and it's suggested in the literature that sometimes heavy metals can change the bacterial community. So does that have human health outcomes outside just the direct effects of heavy metal contamination? So lots of questions that we can learn from this project with our network and with a really cool ecosystem. Okay, so we've got a cool project. We've got some cool questions, but how do we actually execute this project? So let's walk through it. So first off is we want to make sure that everybody has the supplies that they need. So top left here, you can see us packing up a bunch of BIODIGS sampling kits. This is us taking over, maybe a little worrisome for the undergraduate teaching lab. We kind of took over the space there and started packing up boxes of buckets and shovels and reagents and tubes and all that good stuff, and sent them out to our collaborators across the country. And you can already see some students going ahead and preserving some soil with a DNA RNA shield preserving solution just to make sure we keep stable everything that's already in the soil. Okay, so a little bit more detail. Should mention a fun part of my job is applying for the permits. We want to make sure that we're doing things totally ethically here. There's a lot of things we might want to consider when thinking about the autonomy and sovereignty of land. We want to make sure that people are okay with us sampling soil and understand kind of what we're going to do with it and are okay with that, as well as making sure that moving soil across state lines is done in an ethical way. I have an old friend who studies plague out in Colorado, and he found that these plague cysts can persist in the environment for a really long time. So we don't want to inadvertently introduce something that could be potentially pathogenic or hurt somebody. So want to be extra careful. We'll go ahead and select a bunch of individual samples in a given area, not too big, about a 10 by 10 meter area, because we don't want just a single point of soil. We want to get a kind of representative of the area sample, and we're going to homogenize that into a composite sample. We'll collect some metadata, snap a photo, GPS, field notes, good stuff like that. And so we'll end up with 2 DNA samples and 2 a bit larger bulk samples for understanding the soil physical properties. We'll repeat that across 4 additional sites, and these could be sites that are interesting to the faculty that is doing the sampling. So we had some faculty that were interested in sites that historically had been industrialized. So there were some heavy metal, potentially some heavy metal contamination. They were very interested in that. Had others who were interested in agriculture. We had one faculty who knew somebody with an organic wine farm and really was curious about the soil there. So they went out and did a more agricultural site. So After we've got these samples all collected, we're going to bring them back to the lab and do some soil testing on them. I mentioned heavy metals. We're going to detect those, so things like lead, cadmium, arsenic, as well as some other elements and organics and pH and things like that. And then the exciting thing, we got the DNA sequencing of those sample metagenomes. After we have the data in hand, we're going to analyze that on our favorite cloud platform, Anvil. As David mentioned, I'm a PI of that, so I do want to make sure we use the platforms that are accessible and can be used regardless of whether you have a computing cluster. And then using our favorite tools, Galaxy and Bioconductor, to do the analysis. All right. So our BIODIGS scholars are actually already hard at work on the analysis. So on the top left, you can see some work that was done in Anvil, just looking at some K-mer analysis to compare samples that we would expect to be similar. But down here is a really cool thing I'd like to share. So this is a Galaxy workflow that a student, Catherine Ulbricht at Spelman College, developed to understand taxonomy and function in some of the samples. So you can see kind of the reproducible workflow that you can follow along here. And Catherine Ulbricht and Nia Davis actually presented some of their work at the Plant Animal Genome Conference recently. And really amazing. Just learned, I want to say like a week ago, that Catherine and Nia actually won a college-wide science award for their work on BIODIGS. So it's just really amazing to see these scholars already making such amazing breakthroughs and diving into the data and really getting started on their science careers. So just really great to see that. And it's not just data living on Anvil. We've also got open source data that's available. So the BIODIGS package allows you to bring in data into R pretty programmatically and pulls from our most recent soil data. So if we've done some recent processing, you can get the most updated version of that, but that's all open source for folks to jump in and start doing analysis right away. All right, so that's some of the work our scholars are up to, but I did want to share some kind of bigger analysis at a little bit broader scale as in the project as a whole. We did do some pilot analysis and pilot sampling to try and get do some of the troubleshooting of the protocols and things like that. So I want to share a couple of findings that we've come up with that we're just pretty excited about. So first, let's get into some of the genomic diversity we're actually seeing among the samples we've collected. Okay, so I just mentioned that there is that we had conducted a pilot study sampling. So we did send out kits to and collect soil from tons of places, but we wanted to make sure things were, all going well and smoothly before we, sent kits out to folks and then they're kind of left scratching their heads. We wanted to make sure that everybody kind of knew what was up and had easier protocols to follow. So we did a little bit of pilot study just to kind of figure things out a little bit better. So we collected 24 samples from Montgomery County, just outside DC, with students from Montgomery College. And we collected samples around Baltimore. So 24 samples, even collection there, with students from Notre Dame, Maryland University, Montgomery College, and College of Southern Maryland. So we have a little two-city comparison here. And just wanted to mention too, this map comes from biodigs.org. So you can, if you're kind of big on playing with the data and exploring maps, this is all interactive. You can kind of check that out and kind of explore our sampling sites, get pictures of all the sites, and see some soil properties and things like that. So this pilot study, just to give you an idea of what some of the sites look like. So one of our sites was Stony Run. This is near the Homewood Hopkins campus. It's this kind of long, skinny park. Love to go back there and do a little running, but always see people like walking their dogs and things like that. It's pretty popular spot. And you can see this trail right here. So it's definitely got some human touch, but largely unmanaged, right? It's trees and kind of leafy and jungly looking, maybe in some ways. But yeah, lots of trees, weeds, understory, things like that. We also have a site near Gwynns Falls Trailhead. And so this is a historically a little bit more industrialized area. It has, had lots of factories and things nearby historically, and you can still see some of the urban impact. This is 95 right here. Yeah, just a lot of kind of urban footprint at this site. And then a lot of our sites actually looks a little something like Lake Needwood in Montgomery County. This is like more of a suburban park. Doesn't have a lot of urban stuff nearby, but you can see that it has grass that's mowed and taken care of and managed. But there's large trees, there's lakes and things like that too. So all kinds of different ecosystems, some really impacted by humans, probably more these two, and a little bit less so. All right, so from this pilot data, we took samples and stored them and preserved them on DNA RNA Shield, and then brought them to our fantastic colleagues here at GRCF, who extracted them using Power Soil Pro Kit. There's special accommodations you'll need to do to kind of process soil and just make sure you can get the DNA out of it okay. They were able to run a paired end 150 base pair NovaSeq run, giving us, out of those 48 sites, about 20 million reads per site. The quality just looks fantastic. We're really kind of pleased with the results and excited to dive into the data at this point. So the soil testing data as well. So we were able to send this to the University of Delaware. They have a really comprehensive soil testing facility there. So they are able to get us things like soil pH and then individual elements, calcium, magnesium, manganese, things like that, as well as the heavy metals, which we think might have some effect on the metagenomes and the microbial communities that we're seeing. All right, so I want to share some exciting findings in terms of taxonomic classification. So our team was able to do some metagenomic analysis using Kraken-2. And we do find some archaea and some eukaryotes, but largely what we're seeing here is bacteria in the soil. And I want to call attention to a couple different groups here, a couple different clades. So the first is this Nitrobacteriaceae. And this is largely, common soil microbes, nothing too unusual there. But these are organisms that closely associate with, or a lot of them are organisms that closely associate with plants and help them fix nitrogen and gather nutrients from the environment. So really important for keeping plants happy in ecosystems. Also wanted to point out this actinobacteria group. So a lot of the species in this group do something very similar. They associate with plants and help them survive in environments, but really actually pretty important economically for us for environments, natural environments, but also for agricultural environments, helping plants thrive and collect nitrogen. And yeah, I think the actinobacteria and Nitrobacteriaceae, having really common and similar functions there. Okay, I did want to call attention to a couple other groups. So there's several hits to the genus Streptomyces, which produces many of our naturally occurring antimicrobials. This is something that we're very interested in from an understanding of antibiotic and antimicrobial resistance. This is where our new antibiotics come from, is from the environment. And there's also some interesting clade here, Verrucomicrobia. And some species of Verrucomicrobia are actually pretty important for probiotics and for GI metabolism. It's pretty interesting as some of these Verrucomicrobia actually break down plants in the environment. So they're kind of decomposers. And it's kind of interesting. They have maybe a similar role in the environment and inside the human environment. But interestingly, the verruca microbia are actually pretty hard to culture. So if you take a plate and you're trying to assess what's there by streaking your bacteria plate, these guys are a little bit harder to detect. So we may be actually know less about them than we think based on their abundance in the environment. All right. So those Kraken results, very interesting, but we were very surprised to see that quite a large percentage of those reads are unclassified. So we really don't know what the rest of that information is. And there's a lot of unknowns still. And so we're wondering, can we do better? Can we try to figure out what the rest of these reads are doing? And so we were really pleased to find a recent paper talking about soil microbiomes and basically a genomic catalog of these microbiome assisted genome assemblies. So these MAGs, over 40,000 of these, and seeing if this could help us actually quantify some of those reads a little bit better. And we were able to use these with the program SILF and actually got much better classification and determination of kind of what's going on in the soil. So taking a look at the x-axis here, we've got a bunch of different sites. The name's not super important, but you can see on the left is our Baltimore sites, and on the right, DC or Montgomery County, and then relative abundance. And I'm not seeing huge differences spatially, so between the two areas, the two cities, but a couple of kind of interesting things. So I can see a couple of sites, this one here and this one here, have more of this kind of red color. This is the Acidophorales clade. And just the fact that these guys are abundant is pretty interesting. Both of these sites were kind of leafy. Remember that picture of Stony Run that I showed you? They're more reminiscent of kind of like a forest or just a kind of wild area. Versus a couple of sites in Baltimore, got this one here and this one here. They've got an abundance of this kind of olive green clade here. And these sites were kind of interesting because they were both more managed fields. They're mowed. It's basically all turf grass. Places where folks are, playing soccer and baseball and things like that. So it's interesting to kind of see some variety that might correlate with the plant life that's growing there. But yeah, largely we're seeing, a good amount of diversity. A lot of this kind of teal color, this nitrosospirales and nitrospirales groups, important again for helping plants gather nitrogen from the environment. Okay, so here we've got our genomic diversity, but let's explore a little bit more. I mentioned the plants growing there, but let's explore a little bit more about the environmental associations that might be related to the metagenomes that we're seeing. Okay, so first want to call attention to some of the soil data. So the environments that we're collecting our DNA samples from. So we've got iron, lead, and arsenic. We can see that there's a couple sites in both areas that have a little higher than normal iron concentration. There's also a couple sites that have higher than normal arsenic concentration. But I think one of the starkest findings here, and this tracks with what others have found, that lead levels in Baltimore are quite high compared to the surrounding more suburban areas in Maryland, with the exception of Leakin Park, which is this kind of like green oasis over here. And Leakin Park is interesting because it was preserved as like, just this natural area for a wealthy family way back in the day. So it never really had factories or industrial activity on it compared to the rest of Baltimore, which had a lot going on. And sadly, the cleanup of the city and restoration of those soils hasn't really been prioritized. So we still are seeing that legacy today. So very stark contrast. And if you do like to grow your own vegetables like me, just make sure you wash them really well. Okay. So in terms of the communities and how they relate to these heavy metals in the environment, right off the bat, I'm not really seeing any stark patterns. I am seeing some common, these nitrospherales and nitrosospherales groups, the kind of teal blue, tend to be really abundant in all levels, maybe a little bit more abundant in the low iron and low arsenic. But yeah, largely similar communities across these different heavy metals. And when we do some statistical analysis on the community, you know, permutational novas and things like that, we're not seeing any significant changes there. But I did want to call attention to some of the safety limits here from a public health perspective. We actually don't have any safe arsenic cutoff in the US, but the EU limit is 20 parts per million. And we can see there's one site that is over that. So it'd be really interesting to dive into that particular site and explore some of the genes that are present. You know, are they particularly good at dealing with high arsenic levels? And then the lead safety level varies by state, but the Vermont safe limit is 30. And you can see we have many sites that are higher than that. And so again, it'll be interesting to explore specific clades and specific genes that might be linked to those high lead levels. But in terms of our really big, broad picture of clades, we aren't seeing a whole lot of differences at different levels. However, we are seeing some interesting patterns for other elements, and I want to get into this just a little bit. First thing that I noticed was that maybe for calcium and boron, we might be seeing increased abundance of those nitrospherales and spirales as calcium and boron increase. They kind of look a little bit similar here. But really interesting is actually aluminum. And at high levels, or sorry, low levels of aluminum, I see a lot of these kind of teal dots, these nitro associated bacteria clades. And then they seem to decline as you go up in aluminum. And at the highest levels of aluminum, we see different clades being kind of more dominant and more abundant. And so these are the acidophorales and the pteroglobales that are a little bit more abundant here, and can see some significant effects when we run a permanova, and maybe also some interactive effects. But don't want to make this seem super alarming or anything like that. These clades of bacteria, there's nothing inherently harmful or worrisome about them. But the acidophorales are kind of interesting. They're another hard to culture species, so we don't know as much about them. And they can sometimes be associated with high metal content. And at this level, we might be having some aluminum toxicity happening. But in general, these clades are a little bit more associated with nutrient poor and acidic soils. So something that we might see actually in a rainforest that's pretty common to see. And the site is pretty leafy and wet. So kind of interesting to see. And before I move on, I did want to mention, this was like a last minute hot off the press finding, but generally in the literature, we find that as pH gets closer to neutral, so going from acidic to kind of a neutral pH, that we see bacterial diversity increasing. And this is usually based on phylogenetic or species diversity. But at the clade level, we actually see about no pattern, or maybe even the opposite pattern. And so I'm always excited when I see something a little controversial. I'm very excited to dive more into that at the species level and see if we see consistent patterns with pH that track with literature. Okay, and then finally, let's dive into some of the human health and disease implications just a bit. And this is where long reads enter the story. So we're also able to collect some nanopore sequencing data at GRCF, and got some really awesome yields with the nanopore. So this was on 4 samples, a subset of 4 samples that we ran. And also did a little bit of work with PacBio. I can see the sequel results are a little less surprising or exciting here. But actually got some really awesome results with the Revio, some really awesome yields and pretty high N50. So what can we do with this long read data? So here are some of the metagenome assembly results we've got from the different data sources. See on the x-axis is the cumulative sequence length, so just all of the sequences we're able to collect, and contig length, so how long those individual assembly chunks are. And so up at the top, we've got our really long contigs. These represent our complete genomes that we're able to assemble. A little bit lower are not quite complete, but pretty easy to categorize. And then we have this really kind of a long and abundant tail that represents novel genes, plasmids, and chunks of maybe some low abundance species that we're able to detect that we weren't able to with the short read data. Okay, so exploring this Revio data a little bit more, because I think it was pretty cool. We worked with PacBio to do some of this stuff, but they were able to assemble 158 MAGs, so those metagenome assembled genomes, and 55 of those were high quality, and only one could be assigned to a known species. So cool with this different sequencing approach. We're able to basically contribute a little bit more to the field of metagenomics and throw some additional species out there that folks will be able to explore. Interestingly, archaea were highly represented, so that kind of supplements those original Kraken results. If you remember, we only had a tiny chunk that were able to be identified as archaea, so kind of adding to the collection here. And interestingly, we were able to find way more of these MAGs with the long reads versus the short reads, and even 27 single contig genomes, which is pretty cool. So taking those different data sources, we wanted to explore whether there would be potentially any antimicrobial resistant genes present, kind of genes of concern, using this tool called Abricate. And you can see with the Revio data, we were able to detect over 160 of these antimicrobial resistance genes, so a little bit concerning. And as you go from these long read sequences to the short read, we're able to have higher detection power with the Revio and the nanopore there. And so when we break that down, we're actually able to see that the majority of these genes were related to vancomycin resistance, so the VAN-R sequence here, which is a little bit worrisome because vancomycin is considered kind of one of these antimicrobial drugs of last resort, working with a lot of treatment-resistant pathogens and things like that. So that's a little worrisome, and something that we might want to explore a little more, what's causing that and where that's coming from. All right, and this is very preliminary, but I did want to kind of see if we could just think about and imagine some of the connections for human health going forward. So recent evidence has shown that the gut microbiome can have a relationship with quality of life and our mental health and just how we're feeling in general, much more important than we have historically thought it was. And so a lot of neurotransmitters and these really important chemicals that are important for well-being are present actually in greater abundance in our gut. So pretty important for us. And I want to zoom in a little bit on GABA for a moment. We take a look at some GABA pathways. We actually see many of these represented in the soil metagenome as well. So I do think this is, again, just the tip of the iceberg, but it'll be really interesting to go forward to understand kind of the relationship between the soil metagenome and the human gut microbiome and what that relationship looks like. Can we detect connections? And what does that mean for human health and well-being, aside from, just levels of heavy metals and other and antimicrobial resistance genes and things like that? Is the story a little bit more complicated? And so lots of exciting things that our team is going to be exploring soon. All right, so just to summarize, the BIODIGS project has done some really awesome work so far, exploring the interaction between soil metagenomic diversity and human health. It's been an enormous team effort just to get the samples collected, sequenced, and some preliminary analysis done. So this is just the tip of the iceberg, but we've already been able to discover new genomes and genes, discovering some antimicrobial resistance genes present in the soil, as well as neurotransmitter activity, which is pretty interesting. And that we've seen just vast improvements from long reads in terms of detecting these mags that are important for kind of documenting the diversity in the soil. And so something that's really important to me is we're also empowering the next generation of scientists, right? If I can train 5 scientists, right, I have just totally expanded my ability to kind of do innovation and discover new things. So just really important to me that we're able to train some really awesome folks. So we've been able to teach state-of-the-art genomics and data science. Students have already won awards for their work, and provide open access to data and compute for community research, right? This is a huge team effort. We need a lot of hands on this work. And our next steps are exploring dynamics across space and time. As I mentioned, what does rainfall do to the microbial community? What does time do? Seasonality, those kind of things. We want to do more longitudinal analysis, but also involve many more institutions, because I think the more variety we get, the more interesting things we're going to learn. But also understanding other data sets that we can incorporate into the BIODIGS project. So health exposures, climate, additional health data. We have a postdoc that's very interested in exploring the respiratory effects of heavy metals and how that relates to the soil microbiome, as well as additional training materials. I think it's really exciting to train people using this data because everyone can kind of imagine the soil outside their home. All right, and so this wouldn't have been possible without all the incredible students that volunteered their time and excitement to collect these soils and to gather some of this data. So they are the real rock stars here. But also the incredible GDSCN faculty who have really trusted us to help organize this project and have really been super excited about it, as well as GRCF for answering all of my silly questions and helping us out and really being awesome collaborators. I really appreciate you guys. And thank you. Do check out biodigs.org. A lot of cool stuff there. If you're interested in being involved, please, of course, reach out. But we also have some resources on the website you can check out here. So thanks.
Thank you, Ava. That was really interesting. Do we have any questions?
Yeah. So thank you.